My name is Joshua Mugabi. I welcome you to Inheritance and Evolution, Topic 7. We are going to look at a learning outcome. A. Analyze the structure and function significance of nucleic acid in meiosis, mitosis, their role in cellular function, and how mutation in nucleotide sequence can contribute to diseases like cancer. The learning activity, we are looking at learning activity C. In groups, learners watch animation or video and make a write-up or a role play. 1. Semi-conservative DNA replication. 2. Protein synthesis. Present their finding to the class. We are going to start by looking at the semi-conservative DNA replication, and we are mainly going to emphasize on replicating DNA model. Our item 1, a teacher presents a 3 model of DNA with color-coded strands. Blue, the original parent strand. Red, the newly synthesized strand. After the replication demonstration, each new DNA molecule contains 1 blue strand and 1 red strand. Task: identify the pattern shown in the replicated DNA molecules. Explain why only 1 parental strand appears in each daughter molecule. Name the replication model demonstrated. State 1 advantage of this replication method. Let us start with identify the pattern shown in the replicated DNA molecule. The pattern: each daughter DNA molecule consists of 1 old blue and 1 new red strand. 2. Explain why only 1 parental strand appears in each daughter molecule. Explanation: each original strand serves as a template for building a new complementary strand. 3. Name the replication model demonstrated. Replication model is the semi-conservative DNA replication. Four. State 1 advantage of this replication method. Advantage: reduced errors. 1 strand carries correct genetic information for accurate copying. Ensures faithful transmission of genetic information from cell to cell. Now, still under semi-conservative DNA replication, we are now going to look at Mendelssohn-Stahl experiment simulation. Item 2. Students perform a virtual lab simulating the Mendelssohn-Stahl experiment using heavy nitrogen, nitrogen 15, and light nitrogen, nitrogen 14. After 1 round of replication, results show intermediate density DNA. Task: explain why DNA showed intermediate density after 1 replication. Predict the result after the second replication cycle. State what this experiment proved. Let's start with explain why DNA showed intermediate density after one replication. Intermediate density was because each DNA molecule has one old heavy strand and one new light strand. Predict the result after the second replication cycle. Second replication, half of the molecules will be intermediate and half will be light density. All new strands paired with new. State what this experiment proved. Conclusion. This experiment proved that DNA replicates in a semi-conservative manner. We are still in semi-conservative DNA replication. Now we are going to emphasize on a mutation in DNA polymerase. Item 3. A student imagines a cell where a mutation weakens the proofreading ability of DNA polymerase. Task. Explain how this affects semi-conservative replication. Predict the consequence for the daughter DNA molecules. State what normally prevents such widespread errors. Let's start with explain how this affects the semi-conservative replication. Effect. More mistakes occur in the newly synthesized strands. Predict the consequences for the daughter DNA molecules. The consequences are a high mutation rate, genetic disorders, or cell malfunction. State what normally prevents such widespread error. Normal prevention, DNA polymerase proofreading and repair mechanism ensures accurate semi-conservative replication. Now let us change and go to protein synthesis, transcription, and translation. Here we are going to look at from gene to protein in a cell model. Item 4. In a classroom animation, students observe DNA unwinding in the nucleus, messenger RNA forming, messenger RNA leaving the nucleus, ribosome reading codons, transfer RNA bringing amino acids, polypeptide forming. Task. Describe what happens during transcription. Explain the role of messenger RNA in protein synthesis. Describe how translation leads to protein formation. Let us start with describe what happens during transcription. Transcription. DNA molecule unwinds. RNA polymerase builds messenger RNA using complementary base pairing. Adenine with uracil, cytosine with guanine. Messenger RNA carries genetic code from nucleus to cytoplasm. Explain the role of messenger RNA in protein synthesis. Role of messenger RNA. It acts as a temporary copy of the gene and provides codon for the ribosome to read. Describe how translation leads to protein formation. Translation. Ribosomes reads messenger RNA codons. Transfer RNA molecules bring specific amino acid using anticodons. Amino acids link to form a polypeptide chain, which is a protein. Item 5. Let us now look at the broken messenger RNA strand. We are still in protein synthesis, transcription, and translation. Item 5. A student analyzes a messenger consequence that suddenly contains a premature stop codon, UAG, due to a mutation. Task. Explain the effect of a premature stop codon on translation. Predict the effect on the resulting protein. State a possible consequence to the organism. Let's start with explain the effect of a premature stop codon on translation. Effect on translation. Ribosomes stop early. Translation terminates prematurely. Predict the effect on the resulting protein. Effect on the protein. A shortened, truncated, and non-functional protein is produced. State a possible consequence to the organism. Consequence. There are likely to be genetic disorders, enzyme failures, or metabolic problems. Still under protein synthesis, transcription and translation, we are going to emphasize on transfer RNA mismatch. Item 6. A student uses a model showing a transfer RNA carrying a wrong amino acid for its anticodon. Task. Predict what happens if transfer RNA carries a wrong amino acid. Explain why the genetic code normally prevents this. State the importance of amino acid transfer RNA synthetase. Let's start with predict what happens if the transfer RNA carries a wrong amino acid. Prediction. Wrong amino acid is added, leading to the altered protein structure and function. Explain why the genetic code normally prevents this. Prevention. The genetic code is specific and unambiguous. Each anticodon matches only one codon. Correct amino acyl transfer RNA synthetase usually loads correct amino acids. State the importance of amino acyl transfer RNA synthetase. Importance of amino acyl transfer RNA synthetase ensures each transfer RNA carries the correct amino acid and maintains accuracy of protein synthesis. We are still under protein synthesis, transcription and translation. This time we are going to look at the environmental stress and protein synthesis. Item 7. During heat stress, ribosomes slow down and misfolded proteins begin to accumulate. Task. Explain how heat stress affects translation. Describe how misfolded proteins can impact the cell. Name the proteins that help refold damaged proteins. Explain how heat stress affects translation. Effect on translation. Ribosomes become less efficient. Transfer RNA binding is disrupted. More translation errors occur. Describe how misfolded proteins can impact the cell. Impact of misfolded proteins. There is loss of enzyme activity, toxic protein aggregates, and possible cell death. Name the protein that help fold damaged proteins. Proteins involved, we have Chapa-1 proteins and heat shock protein, HSPs. Protein synthesis, transcription and translation, we are going to emphasize from DNA mutation to protein change. Item 8. A student note says that a DNA sequence changes from AAT to AGT. Task. Write the original and muted messenger RNA codons. State the amino acids coded by each. Explain how this affects the protein. Write the original and muted messenger RNA codon. Messenger RNA codon. Original DNA was AAT. Therefore, the messenger RNA will be UUA. The mutated DNA was AGT. The messenger RNA would be UCA. State the amino acid coded for each by each amino acid. UUA gives leucine amino acid. UCA codes for serine amino acid. Explain how this affects the protein. Effect on the protein. One amino acid changes leads to missense mutation, may alter protein shape and function. Thank you very much for participating in Evolution and Inheritance, Topic 7. Our learning outcome A, we are looking at the structure and functional significance of nucleic acid in meiosis and mitosis, their role in cellular functions, and how mutation in the nucleotide sequence can contribute to disease, like, for example, cancer. We have been looking at the learning activity C, where a group in learners watch animation and video and make a write-up or a role play. We have been mainly looking at semi-conservative DNA replication and protein synthesis, and you're supposed to present your finding to the class. If this presentation has been very productive to you, please visit me at Mugabi Joshua Biology on YouTube.